Now that we've learned more about the scientific method, what it is, the steps, what's involved in the different steps, let's go ahead and apply the scientific method to an actual biological example. So the example that we're going to use is, let's say that you are a gardener and you love to grow plants. And you have a couple of different fields where you're growing different plants in those fields. And you observe that in one field, and let's just pick a plant, let's say corn, for example. Could be anything, tomatoes, whichever you want to grow. But let's just say for the sake of this experiment or this example, corn. You notice that the plants in field A are growing more leaves, they're producing more corn. The plants in field B have less ears of corn. The corn's not as high, hasn't grown as tall. And of course, this leads you to the question regarding why is the corn in field A growing more than the corn in field B, right? So again, you're kind of looking around, you're observing, you notice that your field A is really close to your property line where your neighbor heavily applies nitrogen fertilizer. And you start to wonder whether or not maybe some of that nitrogen fertilizer is running off onto field A and maybe having an effect on your corn growth. So after a lot of sort of thinking about, what could be going on, you generate the hypothesis that the corn in field A has been exposed to more nitrogen in the soil as a result of that runoff from your neighbor's property than field B. And that's what's leading to the corn in field A growing taller, producing more ears of corn versus what you're seeing in field B. That's your hypothesis, right? Field A has more nitrogen in the soil than field B. That's why the corn in field A is taller and more ears of corn. So we've got our testable hypothesis, right? It's testable, it's falsifiable. That might be right. It might be the nitrogen or it might be something else. And the last step is now we need to develop our prediction, right? So our prediction is sort of, again, we're going to use that if-then. If the nitrogen leads to more leaf growth, more corn production, the corn growing taller, then, right, the fields that have more nitrogen, the corn plants should grow more leaves, there should be more ears of corn. That very logical. If the hypothesis is correct, then this is what I expect to see regarding my results. So right there, right, we've done the first 3 steps of the scientific method. We've observed, we've generated our question, and we have our testable hypothesis. The next step is what we call the controlled experiment. Right. So now, since we've got our hypothesis, we have to test it. The purpose of the experiment is going to be to test that hypothesis. It's going to provide data that's either going to support our hypothesis or it's going to falsify our hypothesis. We're going to have to say, no, we reject the hypothesis because the data doesn't support it. Now, scientific experiments, when we set these up, we like to sort of have 2 groups. We like to have what's called a control group and an experimental group. And the control group is going to be the group that ultimately is not going to receive any outside sort of influences. We're not going to, with the control group, provide that nitrogen fertilizer. The experimental group, on the other hand, is going to be subjected to that change. It's going to be subjected to that fertilizer. So we've got to sort of have these 2 groups. If we're really going to test, right, we have to have a group without the fertilizer and we have to have a group with the fertilizer. These controls become very important because they provide standards for comparisons, right? We can compare the results of the experimental group where we're applying the fertilizer to the group where we're not applying the fertilizer. Now, this really gets us into what we call variables. And with scientific experiments, there's sort of 3 types of variables that we have to talk about and learn about: independent variables, dependent variables, and standardized variables. So you're going to find that there are specific definitions. So for example, the independent variable is the variable that the researcher wants to test. It's what we want to know. Okay, it's the variable that we're controlling, that we're kind of altering between these two groups. So for this study, what we want to know is whether the application of nitrogen fertilizer has any impact on our corn growth. So the independent variable here would be that fertilizer application. So remember, if we go back to our control group and our experimental group, this group is where we would be applying the fertilizer, right? So this one right here is going to get the independent variable. The control group, on the other hand, we're not going to subject to the independent variable, right? That's going to be set to 0. We're not going to fertilize. We're not going to put any fertilizer here with the control group so that we can compare these two. The dependent variable is the variable that's being affected. So in this case, it would be corn growth, right? How tall does the corn grow? How many ears of corn does each plant produce? The dependent variable is what's being measured or observed in the experiment. So what we're going to measure here is how many ears of corn per plant do we get in the experimental group versus how many ears of corn do we get in the control group, or how tall does the corn grow in the control group versus the experimental group. The last set of variables are what are called standardized variables. Standardized variables are variables that are sort of these other factors that can affect the outcome of the experiment. And so we have to sort of make those the same amongst the groups. So for example, we know that water is important for plant growth. So we have to make sure that we give equal amounts of water to the control group, and we give that same equal amount to the experimental group. If we water one group more than the other, that can lead to our data being inaccurate, right? We won't be able to draw good conclusions if we don't standardize the amount of water we're giving to both. Same thing with light, right? We know that plants need light energy to grow. So if one group sort of receives more light than the other group, again, you will not be able to draw conclusions that are meaningful, nor would they be sort of accurate. So standardized variables, usually when we talk about the independent and dependent variable, they're sort of one thing that we're identifying. Standardized variables, there can be several things that we sort of have to standardize amongst the groups. So we've got our experimental group, we've got our control group, we understand our variables. Now what we need to do is talk about sample size and replicates. So what do we mean by sample size? We need to include multiple test subjects. Okay, this would be our sample size if we're going to have solid conclusions. So you can't have, and again, I know this is my picture, but only 3 plants is a really, 3 plants in the control group and 3 plants in the experimental group. This would be an incredibly small sample size. We would want the sample size to be very large, very robust. Okay, the more the larger the sample size, the more that we feel our data is reliable, okay, that it's consistent. If you only have a sample size of 3 and 2 out of the 3 plants die in the control group, again, you are not going to be able to really draw solid conclusions. So we want a big, robust sample size. The other thing that we want to talk about is replicates. So we want to repeat this experiment several times to ensure that the results that we're seeing are consistent, that we're getting the same thing each time, that they're reliable. There isn't something random that's happening. Important to note here is that we don't expect the results to be exactly the same. Okay, from one replicate to the next, because the subjects that we're using, their responses may be different. However, One of the things you have to understand with your replicates is that we're hoping from those results, we are going to be able to sort of average those numbers, and that when we present the results in things like tables and in graphs, that we're going to be able to see trends, that we're going to be able to draw conclusions from the data that we're collecting. This really gets us into presenting results. When we present the results from our experimental study, from our replicates, we often see data presented in tables and graphs. Tables are pretty straightforward. I bet most students have looked at tables before and utilized them. They're really going to be used to summarize and present sort of that final data. Graphs, on the other hand, most of the graphs that we generate in this class tend to be bar graphs or line graphs. We will generate several graphs throughout the semester. One little, maybe point to remember is that typically when we do graphs, the independent variable is going to get graphed on the x-axis and the dependent variable is going to get graphed on the y-axis. Very important that whether it's a table or a graph, that it includes a descriptive legend, sort of a heading that tells us, this is table 1, and then gives us a little description of what we're looking at in either the table or in the graph. As you get farther along in the semester and you're generating tables and graphs, your instructor will provide more information regarding what they're looking for with the table or graph. But the first lab is going to be a great place to go back and look and get additional information and help when generating tables and graphs. So now we've carried out our experiment, we've got our data, we've summarized it, we've looked at it in both a table, we've looked at it in a graph, and we notice, right, if we're using and following our experiment along here, that if we've got our concentration of nitrogen down here, which is our independent variable, and we looked at plant growth, let's say the height of the corn, we can see that there is a trend here, right, that the more nitrogen, right, the more growth, the lower amounts of nitrogen generate the least amount of growth. So again, once we've got that data, we're analyzing it and we're going to draw some conclusions. Once we've sort of looked at the data, one of the first things we want to do is we want to circle back to our hypothesis. Was our hypothesis supported, or did we reject our hypothesis? The data failed to support that original hypothesis. And again, maybe you don't remember, but the hypothesis was that field A has more nitrogen in the soil than field B. And as a result of that, that's what's led to that more growth. So based on our data, right, if we look at this, it seems that our hypothesis has been supported. Now, once we address our hypothesis, it gives us an opportunity to explain why this might be. We can relate it back to our individual study. We can draw some conclusions. We can even offer future directions in this conclusion part of the scientific method. Once we've done all of that, the last part of the scientific method is going to be report and publish your data. So once we complete the experiment, we've analyzed it, drawn our conclusions, it's important to publish your scientific work so that other scientists can learn from what you've done. Now, the articles and the reports that are published can be published in different places. They can be published online where anybody can submit anything, or I prefer, many prefer, what are called peer-reviewed journals. Peer-reviewed journals are journals that contain papers that have been rigorously evaluated by the scientific community and experts in the scientific community before they're published. When they're published in peer-reviewed journals, they carry a little bit more weight because we know that experts in the field have looked at that. If they don't feel that it's been a very controlled experiment, that the conclusions that were drawn are logical, then the paper won't get published. And so again, peer-reviewed journals tend to be a little bit more reliable than anything that is just randomly published on the internet. Now, that was a very quick go through regarding a biological example. The last part of this lab will be giving you an opportunity to sort of design your own experiment and apply and use the scientific method. We're going to do this by using antacids, which is a type of medication that can be used to raise your stomach pH. They're usually taken by mouth to relieve heartburn. Heartburn is usually a sign of indigestion. So we're going to give you a little scenario about antacids, and we're going to give you the materials you need to test, to really come up with a question, generate a hypothesis, and then test that hypothesis, and to collect data. So one of the things that I'll just mention real quickly here regarding the pH scale, when we look at the pH scale, it ranges from 0 to 14, 0 being very acidic, 14 being very basic, neutral being right here in the middle. So again, you'll get some more information regarding this in lab, and given time, you'll really get to take what you've learned throughout this lab and really apply it. If you need help, have questions with any of this information, please don't hesitate to reach out to your instructor or go down to the tutoring center and visit some of our great tutors that would be happy to help you work through the different parts of the scientific method.